everyone, welcome back. In our today's video, we are going to be discussing about spermatogenesis. Okay, so what is spermatogenesis? The word spermato means sperms and genesis means formation. Okay, so in this video, we are going to be understanding about how the sperms are going to be formed, and we are also going to be discussing about the structure of sperm as well as the hormonal control which is involved in the formation of the sperms. Okay, so the spermatogenesis, which is the formation of the sperms. So what is a sperm? Sperm is nothing but a male gamete. Okay, so it is the reproductive cell of a male. It's as simple as that. Sperm is the reproductive cell of a male. Okay, so the process by which these male reproductive cells are going to be formed is only known as spermatogenesis, and this will include all the events which will start from how the spermatogonial cell are going to develop into a sperm. So what is this spermatogonial cell? The spermatogonial cells are from where the sperms are going to be dividing and coming. Okay, so and then when will the spermatogenesis begin? The spermatogenesis in case of male will begin at puberty. Okay, so what is puberty? Puberty is the age of which a male or a female, okay, any child, the puberty is the age at which a child will attain sexual maturity. Okay, so at the spermatogenesis will begin at the age of puberty for males. Okay, so spermatogenesis can be divided into two stages. Okay, so there are two stages in spermatogenesis. So one stage is called the spermatocytogenesis. Okay, so one stage is known as the spermatocytogenesis and the other stage is known as the Spermiogenesis. So what is spermatocytogenesis? So in spermatocytogenesis, we are going to be understanding about how the spermatogonium is going to develop into a spermatid. Okay, so how the spermatogonium develops into a spermatid. Spermatid is nothing but the non-motile sperm. Okay, so the spermatid is nothing but the non-motile sperm, but later on it will become a motile sperm. Okay, alright. So in spermatocytogenesis, we are going to be studying about how the spermatogonium is going to develop into a spermatid. Alright, and then in the spermiogenesis stage, we are going to be studying about how this spermatid is, which is non-motile. I told, right, this spermatid is actually non-motile, but sperm is motile. Okay, so we are going to be discussing about in spermiogenesis how this spermatid gets converted into a motile sperm or the spermatozoa. Okay, so we are going to be taking a look at these two stages in detail now. Okay, so now we are going to be starting off with discussing about the first stage of spermatogenesis, which is spermatocytogenesis. Okay, so spermatocytogenesis begins with how the spermatogonium or the spermatogonia develops into the spermatid. Okay, so in this step, what I've written is spermatogonia. So from where is this spermatogonia coming from? So this spermatogonia is coming from the primordial germ cell. Okay, so primordial germ cells are nothing but the precursors. So precursors means it's going to start something. It's going to from the precursor cell only another cell is going to be developing. Okay, so the spermatogonia is going to originate from the primordial germ cells which are present in the testes of the male. Okay, so where is spermatogenesis basically happening? You know very well. It is happening in the seminiferous tubule. Already when we discussed about male reproductive system, there I must have mentioned. Okay, so spermatogonia is coming from the primordial germ cell, which is a precursor. Okay, so this spermatogonial cells, it is diploid. Okay, so this spermatogonial cell will undergo mitosis. Okay, so all of you know what is mitosis. Mitosis is nothing but multiplication division. Okay, so if it is one cell will divide into two cell into four cell into 8, like that it will go on dividing. Okay, so this spermatogonial cell which is diploid will undergo mitosis, which is multiplication division, and it will give rise to a large number of cells. Okay, so these cells which are coming from the spermatogonia can be divided into two types, as the type type A spermatogonial cells and type B spermatogonial cells. Okay, so this type A spermatogonial cells have a specific function, and type B spermatogonial cell has got a specific function. So now let's see what is the function of type A and type B spermatogonial cells. Okay, so these are also diploid. So now let's see what is the function of the type A cell. Okay, so the type A cell can further be divided into two, as dark type A dark spermatogonial cells and type A pale spermatogonial cells. So what is the function of, first let's see how are they different. Okay, so the reason why it's called as dark and pale is because of the color of their nucleus. Okay, so if the type A cells are going to have a prominent dark nucleus, then it is going to be known as type A dark spermatogonial cells. On the other hand, if it is going to have a little pale, little pale nucleus, then it will be called as type A pale spermatogonial cells. Okay, so it's nothing but based on the color of the nucleus which is there in the cells. Okay, and then they also have few functions which are different. So let's see what is the function of type A dark cell and type A pale cell. So the type A dark cells are going to be developing into the spermatogenic stem cells again. Okay, so type A dark cells are going to be developing into the spermatogenic stem cells again. So the work of the dark cells is to maintain the spermatogenic lineage. Okay, so the work of type A dark cell is to maintain the spermatogenic lineage. So you already know that the spermatogonia is only going to give rise to the sperms. So if all the spermatogonia is getting completed into formation of a sperm, then there will not be any more further spermatogonia to develop into the sperm. So in order to overcome this problem, this type A spermatogonial cells are going to be, especially the type A dark spermatogonial cells. Okay, so the type A dark spermatogonial cell will be forming into the spermatogenic stem cell. Okay, so it is going to be a reserve spermatogenic stem cell, right? So that is about type A dark cells. So when we talk about type A pale cells, the type A pale cells have the ability to differentiate. Type A pale cell has the ability to differentiate into the type B cells. So type A pale cells has the ability to differentiate into type B cells. Okay, so now let's see what is the function of the type B cells. So the type B cells are the cells which are going to be developing into the spermatozoa or the sperms. So it is the type B cells which is coming from the spermatogonia which is going to be developing into the sperms. Alright, so this type this type B cell will divide, okay, and then it will give rise to the primary spermatocyte. So this primary spermatocyte is again diploid. Okay, so this primary spermatocyte will divide again, but then here it is going to divide by the process of meiosis. Okay, so primary spermatocyte divides by the first meiosis. So the first meiotic division will happen in the primary spermatocyte. So what is meiosis? Meiosis is nothing but the reduction division. So far we have been discussing that all the cells, that is spermatogonial cells, type B cells, all of it were having diploid number of chromosome. Diploid means it is going to have one complete set of chromosomes. So after, at the end of meiosis 1, after this the cells are going to be having only haploid number of chromosomes, which is half the number of chromosomes. Okay, so the primary spermatocyte will divide by meiosis 1 and it will give rise to the secondary spermatocyte. Okay, and the secondary spermatocyte is actually haploid. Okay, now this secondary spermatocyte will further divide. Okay, so the secondary spermatocyte will further divide. So what division is going to take place here? It is meiosis 2. Okay, so the secondary spermatocyte will undergo meiosis 2 and it will give rise to 4 spermatids. 4 spermatids, and all the 4 spermatids are going to be haploid. Okay, all the 4 spermatids are going to be haploid. So these are the spermatids. Okay, so I hope you understood this. Now till this, from the spermatogonium till the formation of spermatid, all this will come under spermatocytogenesis, which is the first stage of spermatogenesis. Is that clear? So this is about spermatocytogenesis. So let's talk a little about the spermatids. The spermatids are actually non-motile and they are also going to be having excess cytoplasm. Okay. Now, the next stage. What is the next stage? The second stage of spermatogenesis is the spermiogenesis. So, in spermiogenesis, we are going to be discussing about how this non-motile spermatid is going to be developing into a motile sperm. Okay. And one more thing that you need to understand here is, if at the end of, you can see at the end of spermatocytogenesis, 4 spermatids are getting produced. Okay, and all these 4 spermatids are going to be developing into a functional sperm. Okay, so type B, each type B cell has the ability to give rise to 4 sperms. Okay, that is also important question. So each type B cells, each type B spermatogonial cell has the ability to give rise to how many spermatozoa? It has the ability to give rise to 4 sperms or the 4 spermatozoa. Okay, so I hope you understood spermatocytogenesis. So I will repeat it once again. So it is all starting off with the spermatogonial cell, which is coming from the primordial germ cell. So spermatogonial cell, which is diploid, will undergo mitosis, which is multiplication division, and it will give rise to 2 types of cells. One is type A cell and the other one is the type B cells. So the type A cells are going to be divided further into 2 based on how dark or pale their nucleus is. So the type A dark spermatogonial cell will again form the spermatogonial. Okay, so it's going to form the spermatogenic stem cell again. So it is nothing but a reserve of the spermatogonial cell. Alright, and then this type A pale cell has the ability to differentiate into the type B cell. Okay, so this type B cell is only going to be developing into the sperms. So type B cell is again diploid. It will undergo mitosis again and then it will give rise to the primary spermatocyte. So this primary spermatocyte will undergo the first meiotic division and then it will give rise to 2 haploid secondary spermatocytes. Then these secondary spermatocytes will further divide in order to form the 4 spermatids. So how is it dividing? It is dividing by the process of meiosis 2. Okay, so you know what is meiosis? Meiosis is reduction division. Okay, and then at the end of spermatocytogenesis, 4 spermatids are getting produced. Okay, and then the next stage is spermiogenesis, where this non-motile spermatid is going to be developing into a motile and functional sperm. Okay, so now let's take a look at the spermiogenesis. So you already know what is spermiogenesis. In spermiogenesis, we are going to be understanding how the non-motile spermatid is going to get converted into a mature sperm or spermatozoa. Okay, so there are four phases. So there are four phases in spermiogenesis. The first one is the Golgi phase. Okay, and then the second phase is the cap phase. The third phase is called as the tail phase, and then the fourth phase is known as the maturation phase. So now let's see what happens during each of this step. Okay, so first let's take a look at the Golgi phase. Okay, so the reason why it's called as the Golgi phase is because at this stage the activity of Golgi is actually intensified. The activity of Golgi apparatus is going to be the maximum. That's why it's called as the Golgi phase. So you all very well know about the Golgi apparatus. Okay, so in this phase, in the Golgi phase, the Golgi apparatus is going to be producing few enzymes. Okay, so these enzymes are going to be in the acrosome. Okay, so these enzymes are only going to be forming the acrosome of the sperm. Okay, so these enzymes are basically called as the sperm lysin. Okay, it's known as the sperm lysin. So when we discuss about the structure of the sperm, there I will tell you what is the function of sperm lysin. For now you understand what are the enzymes which are produced by the Golgi apparatus. So all the enzymes which are produced by the Golgi apparatus are going to be hydrolytic enzymes, and then these enzymes are also known as the sperm lysin. Okay, and these enzymes are only going to be forming the acrosomal part. Okay, so the acrosome is going to have the enzymes, have these enzymes. Okay, so the enzymes which are produced by the Golgi complex are hyaluronidase, and then proteinase. So these are the two enzymes which are going to be present in the sperm lysin. Okay, so that is about the Golgi phase. Then the second phase is called as the cap phase. So in this cap phase, the acrosomal cap is going to be formed. So the acrosomal cap is going to be formed. And then one more important point is that the head of the sperm is going to get its shape. Okay, so you all know the shape of the sperm head is going to be somewhat like this. So the shape of the sperm's head is going to be acquired during the cap phase only. Okay, and then in the cap phase also the nucleus will condense inside the head region. So all this is going to happen in this stage. The acrosomal cap is going to be formed, then the head of the sperm is going to get its shape, and then the nucleus will also condense. Okay, and then the next stage is the tail phase. So what's going to happen in the tail phase? So in the tail phase, the tail is going to be formed. Okay, so you all very well know that a tail is important for the sperm to move inside the female reproductive tract. Okay, so for the motility is something which is very, very important for the sperms. So tail is very important for the sperm to swim. Alright, so how is this tail going to be formed? So we already discussed that the spermatid is non-motile, but then a mature sperm is motile. So how is this tail forming? So this tail of the sperm is going to be formed with the help of distal centriole. So distal centriole, the tail is going to be formed with the distal centriole. So actually there are two centrioles which are present inside a sperm. One is the proximal centriole and the other one is the distal centriole. The proximal centriole will be present near to the head region or near to the nucleus. That is why it is known as the proximal centriole. And distal centriole is present little away from the from the nucleus. So it is known as the distal centriole. And the work of the distal centriole is to form the tail of the sperm. Okay, so we will discuss about proximal centriole when we talk about the structure of the sperm again. Okay, so in the tail phase, the tail is going to be developed. How? It is with the help of the distal centriole. Okay, so you all very well know that sperm is actually a cell. So you know cell are going to have all these things. Cell organelles are going to be there. Centriole is a cell organelle. Golgi apparatus is a cell organelle. So all these are not coming randomly from somewhere. As it is a cell, all these things are there inside the sperm. Okay. So centriole is there inside the sperm, Golgi, everything is there inside the spermatid. And later on, it is all maturing and performing different functions. Okay. So moving on to the last stage, which is the maturation phase. So in this maturation phase, the sperm is going to be getting completely mature. Okay. So in the maturation phase, the excess cytoplasm. So the excess cytoplasm which was present in the spermatid is going to be removed. So whatever excess cytoplasm was present in the spermatid, that is going to be removed. How is it going to be removed? It is going to be removed with the help of the Serotonin cells. Okay, so you all very well know what are Serotonin cells. Serotonin cells are the cells which are going to be providing nourishment to the developing sperms. Am I right? So the excess cytoplasm which is present inside the sperm is going to be removed by the Serotonin cell, and thereby the sperm will attain its complete maturity. Okay, so at the end of spermiogenesis, a complete sperm will be formed. So I hope you understood what is spermiogenesis. Okay. So now let's take a look at the hormones which are involved in controlling the spermatogenesis. So we have already discussed that the spermatogenesis is going to begin in male at puberty. Okay. So at puberty, one hormone level will increase, and that is the GnRH. Okay. So what is GnRH? GnRH is gonadotropin releasing hormone. So this gonadotropin releasing hormone is going to be produced from the hypothalamus, and this production of GnRH will occur at puberty in case of the males. Okay, so this GnRH, which is gonadotropin releasing hormone, which is coming from the hypothalamus, will act on adenohypophysis. So what is adenohypophysis? Adenohypophysis is nothing but the anterior pituitary. Okay, so GnRH will act on the anterior pituitary, and then it will make the anterior pituitary to release the GTH. So what is GTH? GTH is gonadotropin hormones. So there are two gonadotropin hormones. One is LH and the other one is the FSH. Okay, so FSH is known as the follicle stimulating hormone. Okay, and then LH is known as the luteinizing hormone. So this GTH are the two hormones which are follicle stimulating hormone and then the luteinizing hormone. So luteinizing hormone is also called as ICSH. So I've already told you in the previous video what is ICSH. ICSH is interstitial cell stimulating hormone. Okay, so now let's see where is the FSH going to act and where is the LH going to act. So FSH is a hormone which is important for spermatogenesis to take place. Okay, so FSH will act on the Serotonin cells and it will make the Serotonin cells to produce what is known as inhibin. Okay, so we have already discussed what is the function of inhibin. So inhibin will be released when the sperm count is high. Okay, so when the sperm count is high, there is no need for further production of sperms to take place. So in that case, the FSH will act on the Serotonin cells and it will inhibit the production, stop the production of the sperms. Okay, so that is one function of the follicle stimulating hormone. Apart from that, FSH is also needed for spermatogenesis. Okay, then the second function is that LH. We are talking about LH now. LH, which is also known as interstitial cell stimulating hormone, will act on the Leydig cells. Okay, so Leydig cells are the interstitial cells which are present in between the cells, and it is in the seminiferous tubule. So the Leydig cells are very important for the production of the hormone testosterone. Okay, so testosterone is again one more hormone which is important for spermatogenesis. Okay, so this is the hormonal control which is there for spermatogenesis. Now let's take a look at the structure of the sperm in detail.